Alrighty, this is Chapter 9, Hearing, Physiology, and Psychoacoustics, Part 2, pages 268 to 275. Starting with the inner ear, it translates waves into neural signals. The cochlear canals and membranes. To begin, the cochlea, by definition, is a snail, tiny coiled structure embedded in the temporal bone of the skull, approximately the size of a baby pea. Filled with watery fluids are three parallel canals: the tympanic canal, the vestibular canal, and the middle canal. The tympanic and vestibular canal are connected by a small opening called the helicochrina, wrapped around the middle canal. The three canals are separated by two membranes: the Reisner's membrane between the vestibular and middle, the basilar membrane between the middle and tympanic, and made up of fiber at the base of the cochlear partition, which is a complex structure through which sound waves are transducted into neural signals. Vibrations through the tympanic membrane to the middle ear bones cause the stapes to push and pull the flexible oval window in and out of the vestibular canal at the base of the cochlea. Movement of the oval window causes waves of pressure to change, traveling waves to flow through the fluid in the vestibular canal. The pressure is relieved from the round window membrane. Vestibular and tympanic cells are wrapped tightly around the middle canal, so when the pressure builds up, it creates bulges. The organ of Corti. The organ of Corti is the movement of the cochlear partition are translated into neural signals along the top of the basilar membrane, scaffolded on the cells of the support, specialized neurons called hair cells, dendrites of the auditory nerve fibers that terminate at the base of the hair cell. The hair cell is in 4 rows down the length of the basilar membrane. In the first row, in the inner row, is 3,500 hairs, and all rows combined is 10,500. Minuscule hair-like bristles called the stereocilia on the inner hair cell are arranged from short to long. Then in the outer, it is in a V or W shape. The tectoral membrane extends at the top of the organ of Corti. Gelatinous flap attaches and they end and it floats above the outer hair cells. They are embedded in it, and on the outer hair cells are embedded in the gelatinous flap that's only attached to one side. This makes sure it can shear movement. In the inner and outer hair cells, pressure equals neural firing. Deflection of the hair cells in the stereocilia, change in the voltage potential, initiating release of the neurotransmitters. They encourage fire by auditory nerve fibers, dendrite synapses on hair cells. Each stereocilium connected by each other is by a tip link, a tiny filament. The stereocilium open ion pore in tiny fraction of a second. Potassium K+ flows rapidly into the hair cell. Rapid depolarization sets in. Rapid influx of calcium ions, Ca2+, initiates the release of neurotransmitters base of the hair to formulate dendrites of the auditory wave. The opening of the pore link is the MET, mechanical electrical transduction, speed and sensitivity of the hair cells, sensitive of 1 nanometer diameter. Then we move on to the coding of the amplitude of the frequency in the cochlea. So the amplitude of the sound wave is increased, and the tympanic membrane and oval window move further in and out with each pressure and fluctuation. The result is that the bulge in the vestibular canal becomes bigger, which causes the cochlear partition to move further up and down, which causes the tectoral membrane to shear across the organ of Corti more forcefully, which causes the hair cells to pivot further back and forth, which causes more neurotransmitters to release, which causes the auditory nerve fibers to fire reaction potentials more quickly. That means that the sound wave amplitude is conveyed in a much same rate of neurons that communicate with the brain. Coding for the frequency is a bit trickier, and so in the book it explains that the cochlear partition is displaced up and down in different places along the basilar membrane. And then, because of the different parts of the cochlear partition, they are displaced at different degrees of different sound wave frequency. High frequencies cause the largest displacement closer to the oval window near the base of the cochlea. Lower frequencies cause displacements further away and near to the apex. In other words, different places on the cochlea are tuned to different frequencies. This tuning is known as the place code for sound frequency. Cochlear tuning to frequency is caused in large part by the differences of the structure of the basilar membrane. The basilar membrane is thick at the base and becomes thinner as it widens. While the basilar membranes get thinner and wider along its length, the cochlear separates frequencies like an acoustic prism. Higher frequencies affect the narrow, stiffer regions of the basilar membrane near the base move, and lower frequencies cause greater displacements in the wider, more flexible regions near the apex. In addition, this means a structural way of being tuned to frequency other process actively sharpening tuning. Remember that there are two different types of hair cells, inner and outer. Over 90% of the afferent fibers of the auditory nerve fibers that take information to the brain synapse over 3,500 inner hair cells, and the inner hair cells are conveying most of all the information. The sound waves in the brains. It turns out that most of the nerve fibers, the synapse of the outer hair cells, are efferent fibers, conveying information from the brain. When these efferent fibers become active, outer hair cells, which have synapses, become physically longer, making them nearby cochlear partitions stiffer. By making some part of the cochlear partition stiffer than other parts, the outer cells make the cochlea more sensitive and more sharply tuned to particular frequencies. Moving on to the auditory nerve for the last subject. The auditory nerve is the response of an individual AN fiber. And the AN fibers are from the cochlea to the brain. Auditory nerve fibers. The auditory nerve fibers, two different frequencies should be related to their place among the cochlear partition. Then the individual auditory nerve fibers in animals, they find that different fibers selectively respond to different sound frequencies. Threshold tuning curves are increased firing to only a very strict range of frequencies. There's a graph. And then characteristic frequency is when the frequency that increases the neuron firing rate and the lowest intensity. The auditory nerve fibers continue to fire at their spontaneous rates as long as the brain knows which auditory nerve fibers have characteristic frequencies. The brain can interpret the pattern of the firing rate across an auditory neuron to determine the frequency of any tone. It's not that simple, the book describes. It is more complex, so you have to consider two types of complications, which is mechanism and low frequency components of sound waves.